Welcome to all of you. Welcome to our distinguished guests. Ladies and gentlemen, Bridgitta, CEO of Fertilizer, welcome to all of you. Before I continue, could I just ask that you stay seated and just for a short while give thanks to the privilege of being here today. Just where you are, if we could just bow our heads for a short period, give thanks. It's an absolute privilege to be here this morning. I'm sure you will all agree. Thank you. Thank you, ladies and gentlemen. Okay, we're quite a large group today. When I put this program together, I thought we might get 30 or 40 people or delegates. And if you look around, the first thing I'd like you to do is, I'm a kind of an informal type person, as you would have gathered by now. Introduce yourself to the person next to you, to the right of you, behind you. Say hello. Get to know a few people around you. Fantastic, thank you. Let's see you introduce yourselves to most everybody. Remember, today is all about networking, sharing information, and making the day a success. Today really is about Rhizobia, and I want to give you a little bit of background. About 2 years ago, working with our insurance company, the gentleman came to me and said, Tom, you know, if things are going to continue with claims working out like they are, we as the industry are not going to be able to insure Rhizobia and or industry. Yeah, I see their heads nodding, quite right. Now, if you think what the ramification of that particular point is, there were claims against Rhizobia companies to the tune of around 34, 35 million, which involved some 40 farmers. Now, please remember, ladies and gentlemen, the success of a farmer really is that crop that's on the land at that time. If that fails, that farmer's no longer there. And you all know, in terms of agriculture and farming, how tough it is not to be able to be insured and lose your livelihood over something like Rhizobia. That makes things rather difficult. Then I thought, but hang on a sec, I can't go to the back of the equation. We really should be looking at the front side. In other words, Rhizobia, what are they? Where do they operate? What is nodulation? What are the factors that affect nodulation? What enhances nodulation? And then I decided, you know, we should put a program together where we can go from A to Z. Now, please remember today, it's very simple. If you do not ask the questions that you need to ask, you're all in agriculture, please ask the question. Because if you're not going to ask the questions, believe it or not, you're not going to get the answers. You need to walk away today with more information to help the industry to be a better success. If we lose any element when it comes to Rhizobia and or nodulation, it's going to be a problem. If you consider that Rhizobia alone is responsible for an increase in yield to the tune of 1.5 tons per hectare, think about what the impact would be if we lost that and we lost insurance and we could not continue along that line. This would be a serious issue. This is why I put this program together, and thanks to a number of people that willingly came to the party to be here today to address you. So, quick round of applause, thanks to those guys. So please, van julle is nie heeltemal Engels magtig nie, ek ken die storie. As jy nou nie in Engels wil vra asseblief, vra dit in Afrikaans, ek sal dit probeer vertaal, en of Pieter sal my help. Maar as jy nou nie vra nie, dan steel jy vir ons en jy steel van jouself. So please guys, Engels, Afrikaans, maak die saak en vra die vraag. Okay, dit is baie belangrik. Maar die inligting wat hier ingewin kan word, is eintlik van groot toepassing. Okay, without further ado, we've got the press here today. Welcome to you guys as well. We've got some very important people here as well from Grain SA. Guys, talk to each other. I know many of you, but really today, enjoy the day. And without further ado, I would like to invite Professor Karin to come and do her presentation.
Good morning, everybody. You speak up because pretty well coming from Stellenbosch University. It actually helps a little bit. So I'm going to keep my fingers crossed that the slides has arrived here in one piece. I think I should just set my timer because I seem to ramble on and on and on. But it is a subject that I really, really enjoy talking about. Just a bit of background, I am a microbiologist by training. I'm actually a microbiologist by training. And my interest in soil microbiology comes from the fungi in soil. And it was purely an academic project. We were looking for penicillin species in famous sites. Because we had a hint that they were doing something in the world. After about 3 years of doing fungi in soil, we realized that there are bacterial communities and these things are talking to each other. And it's actually a lot more complex than we anticipated. So I thought today what I would do is just talk to you a little bit about the soil and the soil environment. Currently I'm working in the regenerative agricultural space. And the best technology that we at this point can afford to look at soil microorganisms. So I hope I can teach you something today. At least I know I'm doing today a lot. So when you speak to people, the first thing that they ask you is what is soil out here? It's just one thing that's going to change my science. Thank you so much. And it actually all depends on who you speak to. I think the best definition that I currently can come up with is this one, which refers to soil health as soil quality, or the capacity of the soil to function. So when we talk about soil health, I would rather use the words functionality than soil health, because soil health is such a loaded term to use. Just coming back to soil, we have quite a bit of land, but just only 11 or 10, 15% of that is arable soil. And we all know working in agriculture that most of our soils are very low in organic matter. And that organic carbon tends to cause lots of problems because that is what our microbes need to live on in the soil. So we have a number of limitations. We have shallow soils, we have extreme textures with rocks, and we just spent a week at the Northern Cape last week speaking to farmers that actually farms in rocks. And very interesting, the first thing that they do when they prepare soils is they go in with the dynamite, which I thought was quite extreme. Seems to work. But the soil that they create in the process is just absolutely phenomenal to see. We do know that most of our soils are not managed very well, but there are an awareness now that we need to take care of the soil, we need to take care of the microbiomes in soil. Okay, so most of our farmers now know that yields are stabilizing and just aside of it, I'm not going to concentrate specifically on soy and rhizobium, but there's going to be a lot of it in there. The profits has gone down. We did not know, we've read up a little bit, but 80% increase in costs in 2022 in terms of input costs, and 90% of our chemicals are imported. Now, in the last week, that has come again. I've thought about what this absolute fluctuation around dollar is doing to the cost of the farmer. It's a very volatile space to be in. So we have more and more farmers that go, oh, we do have this whole legion of organisms in our soils that can actually work for free. We just need to take care of them. We know there's about 10 trillion bacterial cells in every gram of soil. Please remember that number. 30% of the good soil consists of fungi, and they have numerous functions in soil. And many of these can fix nitrogen, and they can mineralize the nutrients for your plant. So they're critical in maintaining soil health. In regen agriculture, this is what we try to do, is to take care of that microbiome in the soil so that it can function and work. Regen agriculture makes it only the pillars are reduced with no tillage. No tillage actually helps that microbiome to stabilize over time. The use of cover crops, and I'll talk a bit about this later. We use composting, increase crop diversity, which increases microbial diversity. If I have time, I'm going to show you some of our results. Increase organic matter, the use of biopesticides, biofertilizers, as well as integrated animals. Microbial communities in soil have numerous functions, and the more we learn about these organisms, the more functions we find. I just want to highlight the most important ones for me is decomposition in soil, decomposition of organic matter, and I heard a lot of my colleagues a while ago say that if it wasn't for fungi, we would be almost hip high in our own waste. So these things are crucial for getting rid of or decomposition. And then also the mineralization and uptake of nutrients. What is very clear is the more we know about, the more we learn about these organisms, the more we realize we actually don't know what they're doing. We have no idea what these things are doing and how they function. For a large number of them, they are unknown to us. We don't have cultures, we can't put them on plates, we can't study them in the lab. And all we see is that what they left for us, a little bit of DNA in the soil. So that's what we have. We make lots of inferences, but the picture is becoming clearer. What we do know is their function in soil, in terms of the microbes, not compartmentalized. These things work in consortia, so they never work on their own. They always have a number of others that work with them. It's actually quite noisy in soil. These things communicate with each other. They can excrete peptides. They talk to each other. They can shut communications down. They produce all kinds of compounds to suppress growth or to increase growth. So it's a little bit noisy and it's a little bit like war in the soil. It's also not homogeneous. So what I collect in Florida is definitely not going to be what I collect in Stellenbosch. And depending on what your crop or your hectare looks like, it will depend how that microbiome looks like. Now for us, what we figured out is that the more homogeneous your crop, if you have monocrops, that microbiome is pretty stable across the lab. You can sample anywhere, you'll have a very similar microbiome. If you want to diversify that, bring in other plants. So things that influence your microorganisms, the biggest thing is pH, and the other one is water availability. So water availability can move your microbiome and completely shift it from one to a completely different one. And pH, and the other one is plants. And this is what we see time and time again, is that the plant that you plant have the biggest influence on the microbiome that it will attract. So we probably need to now dig into soil. So when we talk about the microbiome, we're really talking about the microorganisms that exist in the rhizosphere of your plant. That's where the function takes place. 10 centimeters away from your root, those organisms does not have a lot of influence on what is going on in the rhizosphere. So if you talk about the rhizosphere, there are three regions. Of course, we have the area of cell division, the one that goes through the soil. It's got a little bit more habitat there for protection. It elongates just behind it, and then the third one up there is the area about maturation. This works good. And this is actually really important. Work takes place from the microbiome. So this is a zone of microbial action. pH increase very incredibly important in this area. And most microorganisms like around pH 5 is ideal for them to function, it's ideal for the uptake of nutrients. They can can manage that. So the plant can manage that by microorganisms, bacteria, in particular bacteria, can excrete acids to maintain that pH balance within within that rhizosphere. So if you look at which bacteria and fungi are common there, so you have those that you guys know, the Rhizobium or the Nodulating bacteria. They tend to form symbiotic arrangements with roots, forming nodules, and these associations are pretty specific. And there's going to be other people talking a lot more about the biology of this. But then you also have fungi, the AF, which is common to many of you. You've heard about the vascular mycorrhizal fungi, which has also evolved to follow this symbiotic relationship. And it's very interesting to know that this is the oldest form of fungi that has ever been found, is the vascular mycorrhizal fungi, and we found them in the roots of fossilized plants. So this is a very, very old symbiotic relationship. So I want you to think very, very small now. So if we talk about the rhizosphere, it's literally millimeters around the root. So if you really look into that rhizosphere, about 40%, 20 to 40% of whatever the plant makes to photosynthesis goes to your root as root exudates. And these root exudates can be sugars, it can be peptides, amino acids, alcohols, lots and lots of different molecules that is excreted into the environment or the rhizosphere, and they serve as number one molecules to attract, so attractants, alternatively as food sources for your microorganisms. Non-symbiotic bacteria in that rhizosphere can also modulate the immunity of the plant. So there's lots of functions within that rhizosphere. And if you have to guess how many, remember 12 trillion cells per gram of soil, there's a couple of grams of soil around the root, you can imagine how many cells you have to your disposal to act as an egg. So how do things go in and out of the roots? So most of you should know this, I assume. We make usually lots of assumptions today, but most of the water they just goes through osmosis. And we've had a lot of questions about how things go across and how bacteria enters and exits cells. So this is a bit of a big piece for me. Anything else, elements and nutrients go through active transport. And with this we mean there is an actual channel across that cell wall and membrane where the cell has to expend energy to get the nutrients across. So energy is really important for the plant. So it expends energy, so it has to make sure it's the correct thing coming over. Similarly, it uses active transport to get those root exudates out. So it is an energy expensive task for a plant. And these are the different molecules that would go across. So this is my attempt at being Pixar. So this is what you would kind of see in a very simplified picture of the rhizosphere. So you have the bacteria there, but they're floating around. For those of you who don't know, the charge on the root is negative, the charge on the bacterial cell is negative, so they tend not to be drawn together. So in order for them to get together, they really need to have communicate with the plant, and there are mechanisms in which they do that. And they do this through chemotaxis, which then makes some of our bacteria swim towards the plant and associate with them. We do this by we cut through the bacterial cell. Most of our nodulating bacteria looks like this. They have the front of the cell, they have receptors. The receptors would recognize some of these signal molecules, which binds to the cell. It leads to a cascade of different genes being expressed in the cell, which kind of is just a trigger signal to tell the flagella to start rotating. It looks like a propeller on a ship. If you really go into the biology of these organisms, they're absolutely fantastic how they sense their environment. So we have a directional movement. They don't go straight like my little animation said. So what they do is they sense and then they'll go somewhere and then they will start sensing by spinning around. So it's a little bit like going through a bar when you're a bit drunk. We also have the vascular mycorrhizae here, and there's a very close relationship between vascular mycorrhizae and our nodulating bacteria. This mycorrhizing also depends on the plants for signals, and these are strigolactones, and strigolactones is a compound, a little peptide that the plant excretes to tell the fungus to start making those structures in the cell. So if you don't see that, it will simply not do that. So for those associations to take place, the plant has to tell the microbiome that they need to form that symbiotic relationship. And there are instances where they simply will not tell the cells to do that. So if you just step a little bit back, apart from these direct interactions, we have our nodulating bacteria, the AMF, that is responsible for nitrogen fixation and solubilization of nutrients. We also have indirect mechanisms. And these could be antibiotics, so this is where you have non-symbiotic bacteria and fungi that acts as suppressors for other plant pathogens. And there are numerous ones of them. I think the most famous one that you guys know is Bridgitta. You use it commonly as a biopesticide. We have biopesticide. And it should contain fungi, that's what it does. But it produces all kinds of other things. Mostly it induces the systemic resistance of the plants. So that's what it uses. So there is a case to be made for maintaining a healthy microbiome. So apart from the nodulating bacteria, there's a whole group of other bacteria that can also affect atmospheric nitrogen. These are the free-living or the diacetropic bacteria. And these work in very close association, and I'm going to show you some of the stuff that we did with the nitrogen cycle, and they are very, very sensitive to the nitrogen levels in the soils. So just in case you forgot that I'm a taxonomist, currently these are the nodulating bacteria. They are not just one particular group, there are numerous different genera, they occur in different classes. So the ability to fix nitrogen is not fixed in one group, which I think is very interesting. What is really important to remember is that plants secrete flavonoids that are very specific to certain rhizobia or certain nodulating bacteria. So if it's not compatible, they simply would not combine, or they will not form that symbiotic relationship. So just to refresh your memory, and set the scene for today, nodule formation. It starts with the root hairs. There has to be a trigger for that to happen, and usually the trigger is low nitrogen levels in the soil. The low nitrogen levels in the soil triggers the plant to excrete the flavonoids, which then would recruit these bacteria if they are present. And that's a big question sometimes. So once they get to the cell, they've recognized those flavonoids, they move to the cells, they manage to attach to the root hairs, they would start forming, or the cell would, or the root hair would engulf them. And they do this through a very, very interesting cascade. So we have the flavonoids would go into, so it diffuse into your bacterial cell, attached to a complex called the NOT genes. The NOT genes get expressed as a protein. It forms a peptide that is called NOT factors that goes back into the plant cells, where it links into a NOT factor receptor on the cell wall, which then leads to the cascade for signaling this pathway to start. So this is really important. If those chemicals are not there and are not being excreted and are not recognized, nodule formation will not take place. And then, of course, we have the infection threat. So if everything works out and all of these things happen, then the infection threats will go into the cells, grows, and then the next thing happens. So the NOT genes, the NOT genes there, it's very, very light. It's now been expressed. When that happens, it triggers another cascade of genes in the bacterial cells. And it's called effector genes. And you'll see that my little bacterial cell has now formed a little infection tube into the plant cell. So we have the nodule, NOT factor receptors. We have the effectors that goes through this little infection tube into your plant cell. But also, your plant cell can recognize when your bacteria produces extracellular polysaccharides, which is kind of a gluey substance that it uses to stick to things. So most bacteria that can produce biofilms produce the substance, and your plant cell can recognize that. So now if you have all three of those things there, it would start inducing nodulation. This is the simplified form.
What does happen sometimes, those effectors, if it's non-compatible host microbe, it will trigger a defense response. So that will prevent nodulation from happening. So it can also triggers that defense response. If it's compatible, the defense response is not triggered. And then hopefully, if everything goes well, you have nodules forming and you will be seeing some nitrogen fixated or fixed for your plant. I've noticed there are other people who will talk more about this, but there are various things in the soil that can prevent this. So high nitrogen levels, high salinity, high heavy metals, low phosphorus, and low molybdenum. Let's try this again. Molybdenum. I had it perfect this morning. Will prevent nodule formation. And what happens when you have those conditions, the stress of the plants goes down. As the stress goes up, root exudates goes down, and it recruits fewer bacteria. It likes a nice soil pH of between 6 and 7. Moisture needs to be perfect. And in that case, you will have nodule inflation. And just to show you what the moisture does to your plant microbiome. So a couple of years ago, we looked at honeybush and rooibos, which is also a plant that forms nodules. They live in very, very nitrogen-poor soils, so they are absolutely dependent on these bacteria to fix their nitrogen for them. So what we did is we did high-throughput sequence analysis. So that's basically where we take all of the DNA in a soil sample, we sequence all of them, and we get a list of what's going on. So we can accurately determine which bacterial and fungal species are currently in the soil. So in this case, we only look at the bacteria. And when we compare bulk soils and rhizosphere soils, we couldn't see a big difference between them. So it's very similar communities. We thought that, commercial rooibos and native rooibos, maybe they would have different, but just look at the effect that the plant has. It doesn't matter where you plant that rooibos plant or how you cultivate it, it has its own microbiome. Also no difference between the farms, but if you look at the sampling times, we had a wet season and a dry season, how different those communities are. So we did network analysis, and basically this is just a co-occurrence network. So the larger the circle, the more abundant that group of organisms. And then you'll notice there are some faint lines in there. If it's a dotted line, it's a negative correlation. If it's a solid line, it's positive correlation. So this is during the wet season, highly abundant, a lot of co-occurrence, which is kind of for us an indication of interactions that takes place. And as soon as you go into your dry season, you'll see immediate change in how these organisms interact with each other. And you see some groups becomes really dominant. And in case of this one, it's the acidobacteria, which became very, very dominant. And they are absolutely adapted to grow in very dry, poor nutrient soils. Then remember, your rhizobia also competes with other organisms and native communities. You need some biscular mycorrhizae, which can enhance your nodulation. And just to show you what we mean by this whole thing, so remember we do photosynthesis. It takes CO2, it puts it into the root exudates. Root exudates is excreted, becomes food for your microbiome, and it can also get food from other sources. But when you start adding nitrogen to the soil, you break a very, very fine nitrogen limitation. So you have to have limited nitrogen in native systems to keep your carbon cycle kind of ticking over. If you break that limitation, you speed up your carbon cycle, which kind of changes things a little bit. What happens now is because there's so much nitrogen, your plants does not form those symbiotic relationships and your microbiome becomes competitors. So they compete for the same nutrient sources. In general, not great. This is not what we do when we aim to go towards a regenerative or sustainable agriculture. You'll notice in the next one, the farmer's been nice enough to plant a cover crop there. Cover crops tend to increase diversity in the soils. Again, the same system. We have root exudates as food for the microbiome. Because we're not adding nitrogen, there's a little bit of a nitrogen limitation in the soil. It forces your nitrogen-fixing bacteria to start to fix nitrogen. They will then, of course, form symbiotic relationships with the plants. Mycorrhizae should also go in there if you have phosphorus limitation. really, really important about mycorrhizae is that they are crucial for carbon sequestration. So they produce molecules which is not broken down easily by other microbiomes or other microorganisms, and they can sequester those carbon sources in deeper soil levels. So this is where we're going to. So when people ask you, do I have bad bacteria in my soil? We don't tend to tell them yes or no. Because I don't think there are bad bacteria and bad fungi. And neither are they good ones. They just are, and they survive in the environment that you create for them. The trick is how to manage that. So if you can measure those things, you can manage that environment. And to do that, you have to remember two laws. The one is Shelford's law of minimum or tolerance, which says that an organism has a minimum and maximum and optimum temperature where it can survive. That goes for temperature and pH and food sources and a whole lot of other things. So if you have a product that you're adding into an environment where those things are not optimal, your bacterium is not going to be functioning at optimal levels. The other one is Liebig's law of minimums that states that growth is dictated by the resource in the lowest or the limiting factor. Usually that is nitrogen. So it doesn't matter how much carbon you add to your soils, if you don't have enough nitrogen, you're not going to make any progress. So when we talk about these microbiomes, what you'd like to create within the microbiome is resilience. So you want to have a microbiome that can bounce back, that can protect your plants, and we see this in my diverse microbiomes that are fully functional. They are very resilient against things like drought, for instance. We like to build in some redundancy in there, and this we do with the cover crop so that you have a number of different bacteria living in the soil. So if you, for instance, have a drought experience and you have organisms that do better at drier conditions, they can replace them. But if they're not present, they cannot be recruited. So in agricultural practices, and I've used soybean pictures, I think these are soybeans. If you have a monocrop, you have a very, very soybean-specific microbiome associated with them. Some people like crop rotation, so they chop off their soybean, they leave those roots in the soil. No-till practices will leave those soils and the soil microbiomes intact. You can plant your maize on top of them. Maize, of course, will then recruit their own microbiome, which is very different to a soybean microbiome. It's not the same thing, but you have some overlaps. And as it grows, those microbiomes will develop. Some people go further and they add some crop rotations or intercropping, where you add additional plants there, and you can see that microbiome just keeps getting expanded and getting more and more diverse. And this builds into the functionality, and if you really want to go full regen on people, you add some animals to your system. And we've seen this in some of these systems. As soon as you add animals, it really, really pushes that system to a fully sustainable regenerative system. And what the animals does is, mostly, it fertilizes the environment. It brings new microbes in there, and very important to remember about those microbes. They put all organic nutrients in the soil, but they put in something called necromass. And in terms of the microorganisms soil, that necromass is so important. And it's just the name we give for dead bugs. And it contains a lot of nitrogen, carbon, and phosphorus. So it's a source of nutrients that we don't necessarily measure, but that are present. So it's a very recent paper. I don't think they published it yet, but I got this picture from that shows that as you increase your grazing, almost everything about your microbiome gets better. So I have 2 minutes to just show you a case study we did in the Free State on maize. So what we wanted to do is compare regenerative microbiomes with conventional microbiomes on maize. So we did it over 3 different sampling periods across the season. So this is across the season, fungi and bacteria, and we use the high throughput sequence method to do this. First thing you'll notice, the red ones at the bottom here is the bacterial diversity. It remains lower than the regen farms across the season. So less bacterial diversity. Fungi, not so much. And we think it's because fungi has such a defined function. It doesn't matter which fungi are present, as long as they can decompose, they're there, and they will dominate. So the stack bars here, these are just the different organisms, and if you look at the side there, this is the 50 most common ones, and it only represents about 60% of your microbiome. All of the other bacteria, so within something like this, you would have 1,000 to 2,000 species of bacteria. 50 of them form 60% of that microbiome. The rest of them, very low abundances, but they are there. And they form very, very important functions, even if they're there in low abundances. So size really doesn't matter always. Fungi, exactly the same. And what you will notice, if you can read that list, which I assume you cannot, you'll notice there's a whole bunch of fusariums in there, which immediately got the farmer all up in a tiff. Very interesting, he didn't have a lot of symptoms. And when we started really looking at those fusariums, they turned out to be a number of different species. So the diversity in these fusarium was much better, where in the conventional farm, it was the single pathogen. So if you look at the how these things are put together, what they look like, the red one is conventional, the yellow is one of the farms. So the red and the yellow farm are literally divided by a road. The blue and the green farms, a couple of kilometers away. So where you plant makes a difference, but what you do makes much more of a difference. So we looked at the nitrogen cycle, and what we do is we use a method called qPCR to look at how many copies of a particular gene is present, which gives us an indication of how many organisms are present. So in the nitrogen cycle, we're very interested in this NIFH gene, which is very particular to nodulating bacteria and bacteria that can fix nitrogen. So they need that gene. So if we look at this across the seasons, conventional farms goes down, regen farms goes up. Okay, so sorry, that's me. Stop. Okay, I'm almost done. If you look at that particular gene, this is the gene that microbes use to convert ammonia into nitrate. It's a biological process. Look what happens. So the guy in the conventional farm has really done his full chemical fertilizer path. And they completely shut down that biological process. And I have to add, the other guys also do some chemical nitrogen. It's not like they're not doing anything. There is some cases, but they use it as needed. If we look at the nitrification, again, very low in our conventional farms and picks up. So this is the number of the bacteria that's present during those different times across the season. Then we also look at denitrification, which is also something we worry about. And again, you'll notice that it's quite high in your regen farms, which should raise a couple of eyebrows. But if you up your carbon, that is going to happen. But it is offset if you really look and go into the detail of these microbiomes. These farms also have a lot of methanogens that offset this denitrification. The same story here. And then, of course, another denitrification. Same picture. So, there is no ideal soil community. A plant, you put them in an environment, you create the environment, and they select what they need. And they do this based on their own requirements and the environment they live in. If you're trying to measure these things, please remember that the soil microbiology test should not be used as just a single measurement. It should be done in with a number of other tests, otherwise it doesn't mean a lot. Please remember your soils are very heterogeneous, so sampling matters. And soil diversity is sometimes a lot less important than soil functionality, which is when we do the diversity studies, we always link our functional test with that. So we look and see, if we have this microbiome, does it actually work? Because we have seen farms where they have the most beautiful microbiomes, highly diverse, highly redundant, highly non-functional, because you are just handing them the food. So if you just feed these things and you don't force them to feed themselves, they will be non-functional and become competitors. And with this, I'm going to leave you with a quote from a colleague of mine, said, soil health, like beauty, is in the eye of the beholder. And I would like to thank you for your attention. I'd be happy to ask some questions if you have them. Yes, sir.
Just remember to activate your speakers, please, for the red.
Hello.
I don't have a question.
That was behind you.
Feels like you were getting
I get it a lot. It's because I'm wearing glasses. Let's try this one.
Good morning, everyone. I just want to ask two questions. The first one is based on the statement that low nitrogen levels enhance nodulation. Can you please quantify for me? Because where I come from, most farmers are pushing their yield, so they chemically apply nitrogen.
Yes.
So if you can tell me how much. And then the second question is, I primarily conduct research trials on soybeans, and what I saw this year was that I did not have any nodulation actually two trial sites. And I went to the weather data, and I saw from the 2nd of December until the 24th, temperatures were above 30. There was no rainfall, and there was very poor nodulation. I even called NBFI and BASF people to come and check, because it's an inoculation trial that was conducted on virgin soil. So my question is, I saw that you put the different things that also make nodulation to be poor, and environmental conditions are not there. So I just want to know, do they also play a role, or was it just the soil and the rhizobium that was not maybe done the right way or stored properly, or what could have caused poor nodulation?
Okay. Can I assume you inoculated?
Yes, the
Okay.
trial is a inoculation trial.
Okay.
So we do a control where we plant soybeans. Everything's not fertilized, and we plant them with the control that is untreated. And then I have different products, different names, but the bacteria is Rhizobium. But I have, like, for example, foundation pack, all those Nutrico and all of those products. But it's an inoculation trial.
Okay. So the first question is, it's a difficult one, and I think there are some of my other colleagues may be able to answer that much better. What we found if you start looking at the microbiomes and not just chemical, I'm going to call it chemical farming, the measurement of the nitrogen in soil, the chemical measurement, does not actually tell you the big story. So people look at those nitrogen levels in the soil, and they would either add to the soils, but what we don't measure is the biological nitrogen that's contained in our organic matter. So as soon as people start putting up their organic matter in soil, we don't measure that nitrogen. And I have a colleague in the US who's worked on, I think, maize. He's tried to look at it, but measuring that is incredibly difficult, because the plant has an ability to, if you run us out of nitrogen, you will measure low nitrogen levels in the soil, but the plant has the ability to go and fetch that. If you have living microbes in the soil and they die off, they will provide nitrogen. One of the adaptations that the microbiomes have is to kill off half their populations for nitrogen. So they would literally kill off half the population of microbes to get nitrogen. So there's a lot of biological ways to get it, which we not measure. So it's really difficult to say, if you start working with a biological organism and you use that chemical measurement to correlate that. But if you have high nitrogen levels, I can tell you then probably this nodulation is not going to happen. But I don't know how low should low be. The next question was the temperature. What was the temperature at the roots?
It was anything between 30 to 36.
Yeah.
And that was and it was dry. And
So you probably now putting organisms into a system that's not optimal for them. So they do need the water. So you remember I showed you that they swim. They cannot swim if they don't have water available. So if there's no water at the roots, they cannot get to the roots. That's the way to move around, is through water. So that could be one problem. The other is the temperature. The temperature is kind of at the upper part of where these organisms function. They're okay-ish at 30 degrees. When it gets to 34, 35, they start to struggle. They really like a nice balmy 26 to 28. But we don't have those conditions in the soil, so which is why I'm asking you, below soil, usually it's a little bit cooler if you have coverage over your soils. So and that's the other idea that people have with the cover crops, is to reduce the temperature in soils and to create that cover that would prevent moisture from evaporating. So I think that there could be a number of answers to that question. One is suboptimal conditions. It's very dry. They don't like the lack of water. It's one of the big drivers of the microorganisms is water. But yeah, I hope that answers half your question or a quarter of your question. Yes, sir.
Good morning, Prof. I'm also Afrikaans, so I'll try to use my English before it runs out.
Je kan maar Afrikaans praten, is reg?
Let me try English, I think. So we work with farmers. If you think about the pressures that they starting to get, whether it's regulations or climate variability, the conversation of conservation agriculture, regenerative agriculture is quickly becoming an important topic. And soil health, we try to understand it and be able to guide our farming communities on that. So if I were to listen to your story from this morning, the type of plant that the farmer puts into the ground, first of all, we talk about exudates that it releases that attracts certain types of microbes. So on that thought, there's two questions. Is the microbes already in the soil, or is it brought in through the seeds? Okay. And I guess the follow-up question is, when we talk about now the diversity and the functionality of that microbial community in the soil, then the follow-up question is, the final question is, what can we do to ensure that the functionality of that soil community is optimal?
Yeah. So those are two very, very big questions. So to answer your first question, you don't get soils without microorganisms. And if you look at the recruitment of, if you think about 2,000 species in the rhizosphere of bacteria, the majority of those are present. And I've read a study a couple of days ago. This is really awesome research where they looked at the recruitment of these organisms. So as you move, and they started looking at from 5 millimeter to the rhizoplane, so just that bit, in that little bit of area, they start recruiting there, and it's just generalists. And as it goes towards straight onto the root, it becomes very specialized. So they look into this group and they recruit. Now the specialists like Rhizobia and AMF, when we looked at virgin soils, sometimes they're just not there. And we do analysis for farmers on a fairly routine basis, and we can talk about the analysis if you want to. And sometimes, that is the problem. They send us soils and say, do I have Rhizobia? And we literally sequence everything to find those Rhizobia. So we don't even try and plate them out. We sequence everything. And mostly when they are present in very, very low abundances. So they're not massively big colonies of these things. So it's really important then to make sure that the strain that you have is compatible with your plant and that the conditions are optimal for the plant to be able to send the signal so that the Rhizobium can connect. And also remember, these things cannot swim for a meter. They small distances, so they need to be as close as possible to the root in order for that to take place, because those signal molecules work through diffusion. You need water for that. So if it's a dry environment, you're going to struggle to recruit. That area of recruitment becomes really, really small. And it really limits the chance of finding the correct one. In terms of inoculation, usually in the Free State, we've seen that if your neighbor does conventional agriculture, they will inoculate your fields over his fallow season. So your inoculants come into wind, seeds, composting, the organic matter that you have on your fields, your cover crops, but it can recruit that. So think about, if you look at the bulk soils, there's the sea of organisms. If you plant your plant, it recruits a section of them. Okay. And it's not that species specific for the majority of the microbiome, but it tends to be quite genus and class specific. But it is spread across the bacterial kingdom. Does it make sense? Awesome.
One more question, Karin?
Yes.
So if I can get the right language. Regarding the flavonoids, you
Yes.
mentioned that the plant exudes them. Do we have any idea of the specific flavonoids that are associated with the specific crop? And could we then artificially apply those flavonoids to induce the process? Because I'm thinking if you have
That's a good question.
supplemented nitrogen, but you're not getting nodulation because there's too much nitrogen, but you apply the specific flavonoids or polyphenols, then perhaps you can induce that process.
I think you need to ask the people who really work with rhizobia and flavonoids. Yeah, so I just assumed you got this. So that question will be answered later. My gut feeling is it's going to be tricky. If there are no more questions, thank you very much for your attention.
Karin, thanks very much. That was an excellent introduction to where I wanted to be in terms of the program. The background in terms of rhizobia, the interaction, and Corneille, when farmers start phoning you and saying that there's no nodulation, you can understand the millions of things that need to come together. Where's Corneille this morning? Yeah, there we go. Corneille is a senior person within Grain SA, and we interact quite a lot with Corneille and Grain SA on various levels. Okay, the next speaker is Jonathan. I don't know if I want to embarrass Jonathan, but Jonathan and I go back an awfully long way, Jonathan. That must be more than 25 years. Jonathan is passionate about everything, whether it's his Beechcraft Bonanza, which I think he still has or doesn't have, whether it comes to rhizobia or comes to agriculture. He's the only gentleman I know that in the very early days, when identifying diseases, he'd run around with his laptop. Now, you must know 25 years ago, working with laptops, not really there, but Jonathan was. So welcome, Jonathan. We look forward to your talk this morning.
Thank you, Tom. Good morning, ladies and gentlemen. It's a real honor. Thank you, Tom. Thank you for Fertasa. And it's great to be in, or have this opportunity to discuss Rhizobium. The things that I would like to touch on today is about quality and efficiency of soybean Rhizobium. And hopefully I'll answer some of, or maybe touch on some of those questions as well that we were asked outside. So from MBFI side, I just would like to give you a bit of a history of where soybean inoculants or Rhizobium and the production and the industrialization of that comes from. If we look in the 1980s at the bottom here, right at the bottom here, we see that we started off with non-sterile peat inoculants. In the 1990s, that moved to sterilized peat products. In the early 2000s, products that were developed in the liquid formulations, but at 1 × 10⁹ Then in 2010, in South Africa, we actually had the first formulations of products that contained 10⁻10 bacteria or cells per milliliter. And I think that's where one of the major steps in 2010 As we start seeing, it was almost a decade between each other, we started accelerating that research into the rhizobia, then having, in 2011, developing various different technologies that are like osmolytic technology, signal generation. We're talking about flavonoids now, not factors. How do we actually generate the signals and create those cascades actually happening, and preparing our Rhizobium to actually be able to do that once treated on the seed, and then various other technologies. In 2012, commercializing a pack, and I think as we're going through, we're seeing that we've had to look at this last one in 2018 is optimizing strain optimization in different varieties of soybeans, and making sure that the variety that we're planting is then also strain optimized. And there are variations between that. We did that with Anthony Jarvie with a really great research over a couple of seasons where he did a lot of work on optimization of the strain formulation technology. But then once we find a strain, that strain might not be optimizing for industrialization. So it's quite a difficult and tricky situation where you have the combination of the two situations. Just to give you some, or another one, in 2019, we started our liquid fermentation plant in South Africa. And then in 2022, the same, or just a slightly bigger plant in the US, where we actually manufacture inoculant for the US market as well. So I think there's going to be more and more technologies coming out. The new, or from our side, there's definitely several big new activating or let's say key indicators where we can actually increase the rhizobium efficiency and significantly increase nitrogen fixation. And that's why we've been able to have to research a lot. And I'll show you a bit more about the research now. But I first would like to start off with, I think, a topic that we all not really, well, we all talk about it, but do we really understand this? So I've got on the side here 1 times 10 to the 10 and 10 times 10 to the 10. I'm sorry I didn't change this picture to a fertilizer, NPK, I apologize, but I use glyphosate in my explanation. But we can use nitrogen, NPK fertilizer. So let's take, for argument's sake, a nitrogen source of urea. What do we have in urea? We have a 46% concentration of urea. Okay. If we talk about ammonium sulfate, what concentration do we have? 21. Ammonium nitrate? 28. So, what is the difference between a product that contains 10 to the power of 6 and 10 to the power of 8? A few zeros. Ladies and gentlemen, this is the crux about biologicals. That is 100 times difference. It's not a 10 times. You can't take urea and then make it instead of having, so from a urea to an ammonium sulfate, you have 21% or you have a 46%. That's close to double. 2 times difference. But when we're talking about a biological with a 10 to the power of 6 to the 10 to the power of 7, it's 10 times. 10 to the power of 7 to 10 to the power of 8, another 10 times. So now we're talking about a product that's going from 10 to the power of 6 to the 10 to the power of 8. We're talking about 100 times. And if we go to 9, it's 1,000 times. To the 10, it's 10,000 times. So that you're just trying to imagine if we had to take a product that's concentrated at 1 times 10 to the power of 6, we have to use 10,000 mils compared to a product that's concentrated at 10 to the power of 10, only 1 milliliter to get the same efficiency. Ladies and gentlemen, this is the difference between bulk and nutrition or glyphosate in this specific situation compared to a microorganism. There is an infinite variation in the concentration. And we need to make sure that we are looking at that and being able to provide good quality products that are efficient in high quantity of numbers. So if we had to look at the reason for nitrogen, it's used in every single aspect of the plant. We can look at the from the leaves, the chlorophyll, which is really important for the light intensity and the uptake of, or the use of photosynthesis. When we look at pods, the protein content, plant tissue, the plant growth, and then the roots, obviously, for nutrients and water uptake. So there's a huge importance, and nitrogen is directly linked to the yield factor of soybeans. So the more nitrogen we can get available and make available to the plant, the better our yield will be at the end of the day. If we look at factors that influence or factors of nitrogen requirements for soybeans, we have in our green book from Fertasa and also all the way around the world, a range of about between 70 and 105 kilograms per ton of soybeans produced, or 70 to 105 kilograms of nitrogen required for every ton produced of soybeans. This is very different to maize. Maize needs, if I could have somebody, how many kilograms of nitrogen does a ton of maize require? 15. So we have a very different crop here. We have a crop that requires high amounts of nitrogen compared to maize. And the vastness is very big. So if we had to look at, next one is the one big thing that we would like to, is point number 4. From R1, you're looking at 4.5 kilograms of assimilation of nitrogen per day, per hectare on soybeans. 4.5 kilograms per day. That's huge. So if we don't have the nodules, if we don't have the structure, we don't have the inoculation there and correctly done at that time, we're not going to get enough nitrogen to support our plant growth. The other thing is, in soybeans compared to maize, we're only seeing around 25% of our crop, our vegetative crop, by R1 in soybeans. Compared to maize at R1, we've seen almost 95% of the crop size. So we have a massive vegetative growth stage at the same time as we also needing to assimilate nitrogen for pod production or soybean production. So nitrogen is a huge requirement. And if you look at soybean inoculants, you can fix between 180 to 250 kilograms. These are general guidelines. I've seen a higher amount or through testing. But when we do a registration test, we use a non-limiting factor of nitrogen of 200 kilograms of nitrogen units. So that will equal to close to half a ton, 460 or 480 kilograms of urea, or 720 kilograms of limestone ammonium nitrate, or LAN, which we apply for 1 hectare. So that will be the same as your bacteria treatment on your seed. Okay, so imagine a pallet of LAN or CAR-N, Langsmay, for 1 little packet of inoculant. That's what we've been able to do with the technology. Okay. So, if we have to look at the roots, I'm not going to go into too much detail. Yeah, you did such a good job on that, so thank you for that. But there's so much discussion on this. And how do we actually go from a plant emitting these flavonoids and binding to the membrane of the soybean? I look at it in a different way. I would like to, and soybeans have a specific interaction with the other person or with the crop. So I would like to start off with saying, hello, my name is Jacques. Yeah. So we have to have this happen between our soybean seedling that we put in the plant on our seed and our rhizobia. The better the communication between myself and Stefan, the better the reaction that we're going to be able to cause. Okay, but we need to be able to talk to each other. We need to have the same language. We have to be able to communicate and be able to interact. If we don't have that, we're not going to be able to go forward and actually become and have this mutual relationship. And that's an important requirement. The next thing is, how do we make products or make rhizobia more efficient? So if we had to go and put into a technology, a product where you're having more signals, I'm phoning Jacques now. Okay. Imagine we could put, thank you, Jacques. Imagine we can put into our technologies signals or better communication strategies, get better technologies where we can actually start communicating on more higher technologies, like myself phoning Jacques right now. If we could do that, how much higher nitrogen could we actually create for the plant, and how do we actually be able to, could we increase the yield? And the answer is yes, we can. Through technologies and through developing these technologies, we are able to find signaling molecules and also better communication methods through this structure. Okay. So, a bit of a trick question. Maybe put a jacket on this or something. Okay, but we're looking at primary and secondary nodules, and we're going to get to some interesting information about primary and secondary nodules. So let's say the plant had 20 nodules on it. Let's just say for argument's sake, or 10 nodules. But they all sat on the secondary nodules, or secondary structure of your plant roots, compared to only 2 on your primary root. The same size, but 20 versus 2. What is the difference between the nitrogen fixation of a primary nodule compared to a secondary nodule, or secondary root structure nodule? Anyone? 10 times, 100%. So we can have 20 nodules on the secondary structure and only 2 on the primary structure. And it's identical nitrogen fixation. So nitrogen fixation, nodulation on primary root structure is super important, and that's where we actually start measuring how well our nodulation or inoculant is actually being treated and actually being effective on the plant. Okay, so if we were to look at this slide, for argument's sake, we can see that as the increase in concentration of inoculant, we go from a 10 to the 7 over here, and the third over here, so 10 to the 7. Now we jump 10 times, 10 times, 10 times. And we can see that the nodules around, if you take the entire root structure, hasn't changed significantly. But the amounts of nodules on the primary root have gone from 0.3 nodules on the primary root structure to 7.1. And that means a huge influence and a significant amount of nodulation that increases nitrogen fixation. In some countries in South America, specifically like Argentina, high organic matter, they contain a gram of soil. When they test it, they contain 10 to the power of 8 rhizobia per gram of soil. Our sandy soils in South Africa in the Western Free State or the Northwest, I don't believe that it has even a 10 to the power of 1. But yes, it's a very different condition and a very different situation. Sorry, I'm going the wrong way. So we try to classify what is a good amount of bacteria per seed, and we've tried to use international data as well as our own data, but we've classified this as very good, average, and bad. So anything under 10 to the power of 4 on the seed, it's a really bad inoculation or number of bacteria per seed, and a very good is anything above 10 to the 5, 5 times 10 to the 5. And we do this research on a daily basis. We have a full laboratory that tests this for multiple companies, where we actually do these on-seed treatments, on-seed viabilities for the industry. Oh, I'm going the wrong way the whole time. Okay. Active and inactive nodules. Most of them, I'm sure, have seen a nodule, but active and inactive nodules are really important. Coenzymes, for argument's sake, molybdenum, cobalt, form part of the structure of the nitrogenase enzyme, which then actually forms and helps this nitrogen fixation happen. If we have a brown nodule, we have a nodule that's not fixing nitrogen. And if the darker the red goes, and we've actually been able to test like a light pink, a darker middle pink, and the higher your index of red is, the higher the nitrogenase enzyme is, the higher the nitrogen that we can be able to produce for our plant. Okay, what are important things and features about an inoculant? Research and laboratories, people that are, and HR or people that are well qualified that can look at this on a consistent basis. Because a dilution between myself and if I had to do dilution, and let's say Vilma, where they did a dilution, could be different, but we have to follow same protocols, same procedures, and be consistent through our results. So laboratories and making sure that our consistency is there all the time. The other thing is, if we don't only do just plates when we're testing our Rhizobium, we actually do it on seed in a growing chamber. So every batch or every product will be, or batch of product that's been manufactured will actually be, a Rhizobium test will be done in a growing chamber to identify that the actual nature of the inoculant will fix or make nodules on the specific crop. Sorry, the last one is the Rhizobium is a very, well, I'm going to go through what it entails as far as a Rhizobium is concerned as far as a product. But I think the biggest, and it's a very hard thing with Rhizobium, or to make a sterile biological product, is quite a significant process on large scale. It's quite nice and easy when we do it in a reactor of a 1 liter or 2 or 5 liter reactor. But when we have to take this to a 20,000 or 30,000 liter reactor, the processes that go into that and the amount of backwards or the behind the scenes technology that has to be done to produce that on a consistent basis without contamination is significant. So next thing is trials. We do trials and we trial and we trial and we trial. If we look at the last 10 years, of the last 12 years, we've done over 55,000 test plots in South Africa. That means almost 132 hectares of test plots, not only specifically just Rhizobium, but in biologicals, microbials in a totality. This is a huge amount of in-field data where we're getting it from Bethel all the way through to Pritzker site and all the way in between, which is hugely important to ensure that local data, local conditions, local soils are understood with the product that we're testing. Okay, so what makes a good product? I try to list a couple of things to what makes a good product or what makes a feasible product in the industry. First and foremost, it needs to be registered. We have to go through field trials. We have to go through dossier formation. This will take you several years, but under the CP regulations, chemical regulations, under the L regulations. A product that will contain at least 1 × 10 to the power of 10 at manufacturing. So they could drop in your production, sorry, into your storage over a period of 12 months or 18 months down to 10 to the power of 9, 1 × 10 to the power of 9, but at manufacturing at least a 1 × 10 to the power of 10 formulation. Sterility, either in powder or in liquid, have a very high viable count, and then a long-term shelf life, 12 to 18 months. These are really important, guys. Viability of products can happen. We can make, I can assure you that a lot of people can make a product that lasts 1 month. The thing is to make it last 3 months, to make it last 6 months, or even 2 years almost, is that's the way the challenging side, because it's a growing bacteria. It's not a spore former. The next one is a practical application method. I think this is one of the hardest and trickiest, is because application is a lot of our success in the field. If we don't apply the product well and it's not practical, the success of the product will definitely reduce. Compatibility with either biological or chemical insecticides, fungicides, nematicides, being part of an integrated pest management system, either via seed treatment or via application in furrow. High viability of the bacteria on the seed for up to 21 days. And please, if you guys research, and we get onto Google, we research these wonderful pre-treatment times of the US and in Canada, 120, 180 days. They live in a fridge 90% of the time. We are different. So a product that would have 120 days comfortably in the US or in Canada would have a minimum or a maximum of 21 days in South Africa, if they even get to that high, because their conditions are completely different. Low viable rates that you're applying to the seed or in furrow applications, sterile packaging. This is really, really important. The sterility of the product, the packaging, the actual products that goes out there. Sometimes we actually have these in our bladders. We have them actually swell up a little bit. If they start smelling quite badly, like a spilt milk or spent milk, it means that there's a contaminant that's got through the plastic, because it's actually breathable. It's got little pores in it, and actually is breathable, and it can actually go. It can cause that, but it's not generally when we're making a 10,000 liter reactor or 15 or 20,000 liter reactor of product. 1 or 2 bags would have that through transportation and the breaking of that. Then potential technologies with good strains, with local and international seed varieties, high quality of manufacturing with sterile and constant production. I do believe that growing chamber and highly equipped laboratory is essential, and then large volume of in-field trials to make sure that that product is tested and tested again. Because we have a gentleman that said that we have, a very high, or let's say high temperatures, low soil moisture. Next year you might have great soil moisture, and then it disappears at R1 or R2. And how do we manage that? Because the environment is always changing. But I would like to discuss that more in detail when we have a cup of coffee outside. Okay, so a treatment versus untreated. And this is what we would like to be seeing, is a high amount of those nodules. And if you took a matchbox just under the seed where it's actually been planted from the root base, and you took a matchbox, that's generally where you would find the inoculation that would have happened from the treatment. You could have secondary nodulation, and that would be because of your inflow application. Also, it could also be due to conditions, dry conditions, where you're actually having the rhizobium actually only once it's got water, it's actually infected, but that's at a much later stage. But then poorer nitrogen fixation will happen. But that matched box is what you'll be looking for when you're doing a trial. Treatments, just different treatments in field, and what it actually looks like, where you have the differences between the treatments, or let's say lack of a treatment versus a treatment of nitrogen fixation. I think I would like to, I don't have many more slides left, but I would like to leave you with a thought. Rhizobium is nitrogen in a bacteria form. So if we had to take a production of and uses 750,000 liters of inoculant, we would need to burn 3.2 billion liters of oil to produce 1.8 million tons of nitrogen in a urea form. You guys with it? So we have to burn 3.2 billion liters of oil to convert that into nitrogen fertilizer into 1.8 million kilograms or tons of urea. But via 750,000 liters of inoculant, we can produce the same amount of nitrogen for our legume crops. So we're in the nitrogen business. Rhizobium is in the nitrogen business. We are here to produce high quality nitrogen fixation products, which is going to give you nitrogen, which then feeds your crop. Okay. Thank you for your time.
Are there any questions for Jonathan? I'm sure there are many.
Ja, ek gaan nou Afrikaans praat. Ek weet julle het al genoeg Engelse vrae gehad. Ek sê bang ek kry die antwoord terug en ek moet dit nou met hulle rywele boere gaan verduidelik, so hulle gaan my wegjaag. Okay, ons kry goeie nodulasie, ons gebruik goeie enstof, dankie julle het ook baie goeie enstowwe. Wat kan jy doen om jou nodules langer aktief te hou? Ons weet hier op R3, R4 begin hulle afneem. Is daar enige iets wat ons kan doen om ons nodules langer aktief te hou? En dan, hoeveel nodules kyk julle na op die penwortel vir 'n normale oes van 3 ton?
Ja, 100%. Nee, baie dankie. Ek verstaan dit heeltemal. Ek verstaan Afrikaans heel goed, maar my selfvertroue voor 'n groep mense is nie so goed nie. So amount of nodules, let me go to that one first. That's quite a hard one because it's more about the dry nodular mass. So it could be one massive nodule, it could be 10 small nodules, as long as the primary nodule, the actual nodule is fixed on that primary nodule base. And it's really about a amount of dry nodular mass. And that's actually in milligrams that per plant that you'd actually get. So I would say, as a general rule, from a farmer agronomical point of view, I would be looking at least 2 to 5 main nodules on a plant, on a single plant, that on the main primary root would be sufficient for a 3 to 4 ton crop that you'll be looking for. So going back to your first question, what can we do to increase the shelf life of nodulation or nodules being actively used? As far as we need to be able to increase the solar panel usage. So if we have to, so products for argument's sake like molybdenum, like boron, and also keeping the plant, we've got this situation where the plant is now in a vegetative state, but also in a reproductive state. So where does it put all its energy? And it's a really hard thing to keep those nodules carrying on past R3 to R4 because they start senescing, because the plant is almost finished using them. So it also depends if we can use different biostimulants in that later part of the is really good. But then also using, for argument's sake, various fungicides with a stray, let's say a stay green or reducing of ethylene, that will also increase the nodules quite significantly. But I would say using of fungicides, either biological or let's say a bacillus or a strobilurin, or to keep that activity of ethylene suppressed as much as possible. Molybdenum and cobalts will be your two major indicators as far as longevity of your nodules, and you can keep them a bit longer. If you do face a very, very dry time in that period or a very wet time, you're going to have that senescing naturally, and that is a bit of like what we're seeing this year. We're seeing that at R3 and R4, we're actually seeing a lot of funny nodules what eintlik doodgaan because of too much water. There it's a very different situation where you can't, it's an environmental factor. But you can, but with good biostimulants, you can increase that longevity of the foliar nutrition products. Definitely.
Okay, morning. Okay,
Morning.
I just wanted to find out, are there any specific rhizobial strains you use when you're making these soybean inoculants?
Yes, we do, definitely. We have in our culture bank, a lot of different strains, from the SEMA's 5079, 5080, WB74 for South Africa, as well as then all the USDA's strains. We've got, I'm not 100% sure, I'll have to get back to you with that number, but it's in a lot of, in double digits for sure. So there's differences. I mean, for argument's sake, a USDA 442, for argument's sake, is a very cold culturing rhizobium, where a USDA, which has got the most research globally on, is a 110, and that's more in a very warm condition. Generally, the SEMA, and I might be pronouncing it wrong, but SEMIA, 5079, 5080, as well as then WB74 on good genetics for South Africa.
It's SEMA, sorry?
SEMA, yeah, SEMIA. And I mean, we have seen some compatibility issues with, I believe it is 442 on certain varieties, especially the longer varieties, not the shorter varieties, because that's generally used up in Canada or with the 00s up in North America.
Okay, so all of them, the inoculants will be in liquid form? Or? From
our side, yes, the liquid form, you do get powders. Which you can. The problem is with powders is the concentration per gram of product is much lower than in a liquid, and also the mass production of that and the quality control of small powder applications are quite limited compared to mass production in 20,000 liter reactors.
Okay, thank you.
Ek wil net graag hoor, mens kry baie keer knoppies op 'n diepte van self 50 cm in die grond. Sal dit wees as gevolg van die voorkoms van natuurlike rhizobium in die grond? Of kan die rhizobium baie keer 'n bietjie afbeweeg deur die grond? Hoekom sal mense dit kry so ver van die plant af?
So I think it's going to be a combination of, there is definitely natural rhizobium in soils. We're finding it throughout the world, and that's where we get our isolates from. So there's definitely natural rhizobium. The question is, do they fix nitrogen, and in what quantity, and in how, or what value? So, for argument's sake, I know that in the, let's say, for argument's sake, Carolina area, in the more the eastern growing part of the, let's say, the mist belts in South Africa, a lot of people have said that they don't need rhizobium because they can see the nodulation happen. But when there was an intern in Brapa did a really great study over 20 years, where they did a seed treatment where they have, or they did a seed treatment of fresh inoculant or high good quality inoculant of over 20 years on the same block with a rhizobia that is at 10 to the power of 8 per gram of soil, and they got a minimum of 200 kilograms yield response year on year for the last, or for over 20 years. So to answer your first, or the question, I do believe that could be natural rhizobium, but the question is, do they actually fix nitrogen, and what is the effect that they're actually doing? Ek hoop ek antwoord jou vraag.
Baie dankie. As jy rhizobium, ons sit die rhizobium op die put en ons sit dit in die voor. In die oorsee in Argentinië sit hulle verskillende maatskappye of verskillende strains. Is dit nadelig om verskillende een in die voor te sit, een maatskappy sin in die voor en een op die put? Ek weet julle wil almal julle eie al twee plekke hê, maar wat is die voor- en die nadeel van verskillende strains wat jy een in die voor sit en een op die put? Is dit werk hulle te mekaar of werk hulle simbiose?
Ja, baie interessante vraag. Dit kom op baie. So ek dink nie dis nadelig, glad nie. Die groot ding is meer effektiwiteit van daai produk. So if we had to look at, for argument's sake, the same quantity of, let's say, nitrogen-fixing bacteria in one product versus the other product. Let's say they had the same concentration. They have two different strains. One is a 5079 and a 5080. That efficiency will not have a problem. The question would be is if you had to bring that product that you're putting in furrow at 2 times 10 to the 9, for argument's sake, a lower quality product, and then you're doing a seed treatment with a higher quality product, then there will be a difference in efficiency. So try and stick with your efficiency. I don't believe that the strains are competing unless you choose a very cold strain, which we don't really use, or we don't use in South Africa. I've seen one or two products catalogs like that, and we've tested them, but they actually didn't contain and the big one is 442 from the US. Generally, we don't see that in South Africa. I would say that almost every South African company makes use of the same strains. So it's really about the formulation, the quality of that formulation, rather than the. Don't believe that there's many variations in the strains. So it's more the technology that goes inside the formulation. And sorry, maybe just to, I think the biggest thing is Bridgitta, and maybe Brigitte's going to get into some of that. Bridgitta is a very slow-growing microorganism. So it's not a quick one. So any contaminants within your formulation has a massive impact on your actual yield effect that you're going to get. So contamination is a critical thing that we have to make sure that we don't have in the package. But once you've actually opened the packet, the sterile packet that's never seen a non-HEPA filtered air, that we actually store that correctly or use it within 24 hours or as quickly as possible on the seed. So contamination and other microorganisms like a trichoderma is far quicker. It's like almost like if you could say, a tortoise in Bridgitta Rhizobium and a cheetah. Dis so groot verskil.
Yes, Jonathan, ek wil vinnig by jou hoor. Kom ons sê 'n kliënt het verlede seisoen, soos hy nou gesê, soybeentjies geplant en jy kom laat agter. So hulle het eers laat in Desember geplant. Nou kom jy agter hulle het nie nodulasie nie. Nou het ons gaan stikstof gooi, ons het blare van spuit, ons kry reaksie, maar soos jy gesê het, die hoeveelheid stikstof wat jy moet gee is enorm. Is daar 'n manier om later nog 'n bietjie inokulasie te veroorsaak? Kan jy dit deur die pivot sit? Dis eintlik oorplant, nee. Daar's nie 'n ander opsie nie.
Ons probeer, maar dit is nie 'n werkbare stelsel. Jy gaan 'n stikstof moet opsit, ja, seker. Dit is die beste opsie vir jou beste ROI op daai stadium. So ons het 'n kalender, of ons probeer obviously elke jaar se plantdatum is bietjie anderste, maar ons wil ten minste as ons blokke gaan kyk, dis dit 40 dae na plant of na opkoms van daai soyboon, en daai soyboon moet op daai stadium al reeds goeie knoppies gevorm het. En as ons nie in die ooste, so we've got an eastern and western area. If we do it in the east and it's not good at 40 days, we wait another 10 days, and if there's no rain, or if there's rain in that period and we reevaluate it at 50 days, our latest will be to apply nitrogen will be on 60 days post-emergence to get the massive or the reaction that we would like to require.
Sorry, it was just a follow-up question again. Because what happened was at V2, normally you have plus or minus 6 nodules on the primary root. So what happened was at R1, only then I started getting nodulation. Now, my question would be, will I fix enough nitrogen at that stage? I mean, my assumption is if you start fixing nitrogen from vegetative growth, then most of the nitrogen will move up the plant, and at R1, R3, where you need it the most, it's readily available. But if you start fixing it at R1 or R2, how much nitrogen are you going to store for the plant to use it effectively? That's...
I understand. That's a very cool question. Yeah. So there's some really interesting data on that where it actually shows that a plant, a soybean plant, we only see 25% of its vegetative growth or structure by R1. So 25% of its crop, the foliage. So if that crop is sitting at around 30 centimeters, you're only seeing 30%. So that's where, so to create the structure and the foundation you need, and generally that's where nitrogen from the soil or remaining of, or let's say residual nitrogen, the nitrogen fixation, the bacteria is more biostimulant in the early stages. And then from R1, that's when it really kicks in to make nitrogen. So I don't believe, if we look at, for argument's sake, planting in the east and west, in the east, and I talk about the east as basically the N1, anything to the east, N1 from Johannesburg to Bloemfontein, anything to the east of that, generally we have nodulation much earlier, at V2, V3, V4. In the western areas where it's more sandy, we could expect nitrogen fixation or nodules to actually be only present at R1, where we're fixing huge amounts. And the main insurance or the main requirement is from R1 all the way through, because that's when we produce 75% of our crop structure as well as then our entire yield. I hope that it answers. So I don't believe that you've lost out if you're only seeing the nitrogen, the nodules form at R1. So we had that a lot in Delmas, this region, this year, Bethel, all the way through because of the dry conditions. And then all of a sudden we had some rain, the activity came back up and we found the nodules were there.
Yes, and you know why I'm asking that? Because as a farmer, you start panicking.
Absolutely.
And now
Absolutely.
you think, should I pour nitrogen? How much should I pour? Am I really going to have a positive result if I pour that nitrogen? You know, so, and then you factor in the cost.
Oh yes. So that's where you need to talk to your local consultant, and I think it's very important, ladies and gentlemen, is where we do get our guys. We have great representatives in this country that we do work on farmer level, agronomy level, that we can do these inspections. Like you're saying, you start stressing, and that's where we are here to do the support and the backup to ensure that we have a stewardship or good stewardship plan for the product. So that if there is a problem at R1 or R2, that we go in with nitrogen, sufficient amount, that we actually do sort out the problem. Because the yield, and you know, Tom was talking about it just now, you know, when we talk about a yield of 1.5 tons, we're talking about, at, let's say, 8,000 Rand, the SAFX fluctuates quite a bit, that's 12,000 Rand. If we had to go in with nitrogen fertilizer, it might only cost us 1,000 or 2,000 Rand. So rather go and sort out the problem in the field than wait until we actually harvesting and then we have the problem. So yeah.
I have one question for you, and that is, there's some talk about, in terms of soya varieties, some nodulate better than others. Is there any truth in that?
So yes, definitely there are. There's some very big truth in it. We don't plant such a variety of groups of soybean varieties in this country. Generally, a 00 will be a lot more toxic as far as the chemotoxins or the compounds that are produced on the seed, compared to we grow normally a group 4, 5, and a 6, and maybe a 7, class 7 in South Africa. So a lot of work has been done in that regard as far as the activity and the variety versus the formulation or the strain. We did it in broth mixtures. We didn't do it in any special formulations, and there is definitely a correlation. But most of the, let me put it this way: if we had to go to a 00 soybean, class 00, or even into 1 and 2s, there are big differences as far as inoculants are concerned, for sure. But that's not in our country, so we're talking about this is Saskatchewan in Canada, Minnesota's, they're all planting 00s because they have to get a much quicker growing, or they only have a short window of growing time, and very cold. Where we're growing 4, 5, and 6s, and generally those are our Brady rhizobiums that are registered in this country are all fit for that purpose in that. But yes, if you do a strain, for argument's sake, 442, USDA strains 442, it won't be compatible, or it's not as well compatibilized with our varieties in this country.
One last one perhaps, Jonathan, and that is a lot of farmers use what we call withheld seed.
Sorry?
They keep seed back from the precure. And we've seen some problems there sometimes from a number of aspects. Maybe you could just talk to that for a second.
Yeah, so I think brown bags are an industry norm. I think the biggest question is, the higher the quality of the inoculant that you're going to put on, the more you, let's say, the more technology that you are, using. Sometimes you're planting a brown bag, and we've seen it in some cases, not even on someone who's certified sometimes. So it's not really maybe just a brown bag situation, but a good quality germination is very important. So, if we have a germination percentage of 60%, for argument's sake, and we have to plant, we have to plant 100 kilograms a hectare to get 60 kilograms of seed. And if the kernels are quite big, then you're planting at 4,500, 5,000 kernels per kilogram. So it really depends on the surface area that we're treating. And I think that you can go into organic material that's still left within the brown bag and the germination quality. So you're actually spreading your inoculant over a whole lot more than just the seed that we are trying to plant. So I know we try and get the best seed, but there's some seed with more sclerotia and organic matter than soybeans in some cases. And it is something that, and I think if we have to look at registered seed, a good quality seed provider is very essential on soybeans. So not that I don't think that brown bagging is not a good thing. I think it's good if you can control it and you can manage it. And I think anything is good in the way you manage it.
Thanks, Jonathan. Maybe one more question for Jonathan. Great, Jonathan. Thank you very much.
Thank you.
Our next speaker, I think is Kobus, if I remember correctly. Yes. Kobus, yes, we look forward to your presentation.
Good day, everybody. Thank you, Mr. Chair, for the opportunity. I think it's a big privilege for me and for Omnia as well to speak on this platform. I'm going to speak about a very interesting topic, soybean fertilization and nitrogen dynamics. There you go. So a little bit on the content, a small introduction on the Rhizobium and the fertilizer practices, soil rectifications, and then the summary. So fertility on the Rhizobium formation. Obviously, Rhizobium nodulation is one of the most important fertilizer aspects for soybean fertilization. We all know if Rhizobium doesn't inoculate, we have big problems in terms of yield sensitivity, and it's a very intensive process. Obviously, a lot of carbohydrates and sugars is needed from the plant to drive this nodulation from the Rhizobium. So obviously, the healthier our plant is, the more carbohydrates and sugars it can produce. So a nutrient deficiency or a weak seedling will obviously inhibit or negatively affect nodulation. So this is just a few factors that has an impact. Obviously, sunlight is in there and so forth. But I'm just gonna, talk about the fertility side of it. Organic carbon, soil pH, and nutrient status availability, and obviously with that comes enough soil moisture. And I just put in this schematic illustration of soil pH. We all know the importance of soil pH for a good seedling, actually for good plant growth, and that is our nitrogen, phosphorus. You can see, especially on phosphorus, a low pH, where our soybeans are mostly produced, our phosphorus availability can become a problem, and we know it's one of the energy drivers in our plants. Calcium, obviously, at low pHs can become a problem. Magnesium as well, chlorophyll, for producing those sugars, and then molybdenum. It's obviously pH sensitive as well and very important for the nodulation. Overliming can be a problem because then you go into an iron and boron deficiency if your pHs go too high, and we already heard about the importance of boron. Then I'm just going to stand still a little bit on organic carbon and nitrogen availability in the soil. We've seen in the previous speakers that too much nitrogen will hinder or prohibit nodulation. Okay, thanks. So too much nitrogen obviously if we've had too much nitrogen will have a negative effect on nodulation, and influences on nitrogen. Of one of the big aspects of residual nitrogen is the organic carbon. So obviously there's lots of influences on the clay content, tillage, and crop rotation, and some of the big ones is that we don't have control over is temperature and moisture, especially in dryland conditions. So, but the area that we plant our soybeans in obviously dictates that. So temperature in the eastern part of the country is much lower and there's more moisture. So obviously you'll have higher organic carbon than in the western part of the country. So your nitrogen approach, especially to the seedling, will differ in these conditions. And just a footnote, soil amendments like humic acid and fulvic acid we've seen has a positive effect on that seedling to get a strong seedling. All right, this is just a demonstration trial that we did. It's in the western part of the Western Free State, so it's quite arid. It's soils that has good drainage, and it's low rainfall. The rainfall is around about 400 to 450 millimeters a year on average. And you can see it's a very sandy soil. The map on the left-hand side, you can see the CEC is low. It's below 2. It has adequate phosphorus in the soil and adequate potassium. And we put in a fertilizer mix, a 5-4-3-22 liquid fertilizer with humates and micronutrients, 17 kilograms of nitrogen, 13 kilograms of phosphorus, 10 of potassium, and a little bit of zinc. And you can see on this, I don't know if you can see from there, but with fertilizer actually has a little bit more rhizobiums than without fertilizer. So in this situation, a good planter mix actually enhanced the tempo of rhizobium formation because you establish a strong seedling. Here you can see that's a yield difference around about 400 kilograms with fertilizer than without fertilizer. And you can see on the photo on the right-hand side, the soybeans seedlings or the young soybeans is a little bit weaker than the part with the fertilizer. But I think phosphorus, obviously our energy driver. Soybean needs a lot of phosphorus and it can utilize the soil phosphorus very well. But usually we get good significant reactions by adding a little bit of phosphorus in the band for the young soybean. So many factors, I think that's why we get the reaction, because we have so many factors that has an influence on the availability. We saw the pH schematic illustration just the previous slide. Climate, obviously, if you have low moisture, the plant can't utilize all the phosphorus in the soil. Root restrictions, moisture, compaction, nematodes. Here's just an illustration of what nematodes can do to a soybean plant, and obviously of the diffusion uptake with phosphorus is going to be less in this situation than in a healthy plant. This is just a schematic illustration of how phosphorus is fixated by pH, and again I added a table of the P recommendations from the Fertasa handbook, and you can just have a look. Most of our soils is between 20 and 30 milligrams per kilograms of phosphorus in the soil, and it comes down to around about 10 kilograms of phosphorus for each ton of soybean on yield potential. All right. So this is a trial in phosphorus at 3 locations. So it's just to show what the effect of a phosphorus with plant is in 3 locations here in the Eastern Highveld. So you can see 26 kilograms of phosphorus on the first trial was the economical optimum. The second trial we didn't have a reaction, and then the third trial 30 kilograms of phosphorus was the economical optimum. So you can see it's 2 of the 3 trials worked out, and it's a little bit higher yielding, and it's at 3 tons a hectare average, and it's very close to the Fertasa's guidelines. So we've seen that in practical in the field as well. The guidelines has a good correlation with what we see in practical with these trials. The next one, different phosphorus source, high phosphorus status in the soil, and you can see 20 kilograms of phosphorus was the economical optimum in this situation. So a little bit lower than the guidelines for the 2.8 ton, but still in that region, you still get reaction with a little bit of phosphorus there. Then potassium. Sufficient potassium. Soybean can utilize it very well, the topsoil and the subsoil, but the crux with potassium and soybeans is it needs a large amount of potassium, 19 kilograms for each ton, not to produce. The farmer is taking it to the silo, that 19 kilograms. So it's 5 times more for maize than maize for the same tonnage. So if you're going to 3 tons soybeans, it equals to 15 tons of maize. Right, so obviously our management of our soil is then very important. The thing about potassium, it has a high salt index. So if you want to put a lot of potassium in, watch out for fertilizer burn if you put it close to the seed and close to the rhizobium. Okay, this is the handbooks, fertilizer handbooks recommendation for yields ton per hectare. And again, we have high potassium, so you can work around about 10 kilograms of potassium for each ton of yield potential. All right, and this is just a schematic illustration of where the soybean takes out its potassium. The most potassium it takes out, and we've heard it with the previous speakers, is from the R1, R2 stage, obviously up to the R6 stage. So it's a short period of time that a soybean needs a lot of potassium. And it's a lot of vegetative growth and a lot of reproductive growth in that growth stages. So obviously, mineralization can be a problem. Although our soils have a lot of potassium, is it mineralizing it quickly enough? And that's a big question here. So we had 4 trials, 4 locations in the center part of South Africa. So it's Northwest and Central Free State we did our trials. So you can see sandy soil on site 1. Good potassium status, 163 parts per million. 19% of potassium, and we got a reaction with 80 kilograms of KCl band placed. So it's 40 kilograms of potassium. And that's around about the 2.2 to 2.8 ton region. We had a 600 kilogram yield increase. The next site, even more potassium in the soil. It's a more clayey soil, 240 milligrams per kilogram and 21%. Little bit lower yield, not a big reaction on potassium, 50 kilograms. And the last two sites, it was a dry year, even lower yields, also good potassium status. On the one side, 100 kilograms of hectare yield increase, the other side, nothing. So it just goes to show the higher the demand of the plant, the more restrictive or more, the more the problem is for mineralization and taking that potassium up out of the soil. And if you add a little bit of potassium in high yielding situations, you get a reaction. All right, this is just a demonstration trial one of my colleagues did in the Western Free State. The big thing I want to show you here is it's around about R3 that he made this profile hole, and he took soil samples in each of those blocks on a control and on a treatment. The treatment was 234-30. It was 300 kilograms a hectare that he put in right beneath the plant as a pre-plant, not with plant, as a pre-plant. And I just want to show, before the season, it was 120 to 150 milligrams per kilogram was the soil analysis. In season, it dropped to as low as 70 parts per million. So obviously, it's a high demand from that soybean, and it dropped the soil status. But in the treatment side, that's the right-hand side, you can see right beneath the plant in the second block, you have a spike in the ppms, or the milligram per kilogram of potassium where the fertilizer was put in. And we know it's the fusion uptake, so a high concentration to a low concentration. So theoretically, it should be positive. Same with the phosphorus. We also had a spike right beneath the plant where the fertilizer was put in. And then here's the yield results. You can see we got a positive reaction with putting in this high yielding situation, around about 4 tons. Positive reaction by putting in 150 kilograms of 234 is 290 kilograms of yield increase, and at 300 we got a 360 kilogram yield increase. So especially that 300 kilogram one, very close to the guidelines as well. So we got a positive reaction on that combination of a little bit of nitrogen, phosphorus and potassium there. So definitely in this situation, worth it to fertilize that soybean. Then a very good trial done in the Eastern Highveld. One of my colleagues did it. He's here today. And you can see at treatment 1 and treatment 8 doesn't have phosphorus and potassium in. Treatment 1, no fertilizer. And the rest of the treatments were balanced with 20 kilograms of fertilizer, or 20 kilograms of phosphorus in, and it's band placed with the planter. And then the differences is obviously the nitrogen, and before, even the nitrogen in the mixes and the potassium in the mixes, and as well, and the sulfur as well. So phosphorus is constant except for treatment 1 and treatment 8. All right, here's the yield results. You can see zero fertilization didn't had the worst yield, and our winners there was there in the center. Don't know if my points will show it, but yeah, in the center you can see 20, 20, 20, 14, 21, 28, and the 9, 19, 28. That was the 3 winners in this trial. So what you see is, as the guidelines suggest, around about 20 to 30 kilograms of potassium is a good response. And as soon as you take out the potassium, the yield drops. You can see that 721.0 has a worst, a little bit worse yield than the rest. And as soon as you take out the nitrogen, your yield dropped a little bit. It is a lot of potassium, that one without the nitrogen. So maybe the salt index was a problem there as well. But it looks like as soon as you take out the nitrogen, you had a negative reaction. Then just be aware of your fertilizer placement. As I said, salt indexes from nitrogen and potassium can be a problem. So put your fertilizer away at least 5 centimeters away from the seed, 5 below. And if you want to add a lot of potassium, rather take the potassium out of that mix. We don't want weak plant density or the rhizobium not to inoculate properly. Then a little bit about nitrogen dynamics, a small amount at plant. Top dressing of nitrogen when the rhizobiums is effective is usually not economical. Some guys say under very high yielding situations they got reactions on maybe under a center pivot or so. Double cropping maybe if you put in soybeans right after wheat that has a high carbon to nitrogen ratio. You can get a negative nitrogen negative period, and maybe you can put it in that situation. When Rhizobium inoculant was unsuccessful, we suggest you replant if there's still time. Usually the nitrogen fertilization is so expensive against the amount of extra yield you get. If you have enough time, replant. Obviously, if you plant it late, you have to add nitrogen fertilization. And with your crop rotation, there's obviously a lot of advantages by planting soybeans in your crop rotation. But usually, we suggest you don't cut back on your nitrogen fertilization for your maize that's coming after the soybeans due to the dynamics of nitrogen carryover. And usually, the crop rotation itself gives the farmer a yield increase as well. Right, this is just a trial, a statistical trial that was done with on soybeans where the inoculum was successful, and just to see if this high yield potential soybeans gets a reaction by adding additional nitrogen. And you can see with 20 kilograms of nitrogen at the R1 stage, there was a reaction, but it wasn't statistical. And with more nitrogen than that, no reaction took place. Sulfur, very important element for soybeans because it's an oil, high protein crop, and it uses quite a lot of sulfur, 4 to 8.8 kilograms per ton. And you can, if you look at a schematic graph, you can see 10% yield loss as if you have a nitrogen deficiency, and keep it up to 40 kilograms a hectare in the top soil, top 20 centimeters. So very close to your phosphorus fertilization, you can balance your sulfur. Right, then secondary micronutrients like calcium, magnesium, and your micronutrients, obviously very pH dependable, as I said on before. And liming essential in that situation. Calcium for nodulation, molybdenum, very important in the symbiotic nitrogen binding, and zinc deficiencies. We also get zinc deficiencies, but less than maize because getting your soil in balance, and there's different ways, obviously, to add these elements, but liming is usually the best place to start. The molybdenum seed treatments, early foliar applications with the molybdenum and the zinc can also help with your shortages or deficiencies. Then I think a big one is the soil rectification. We in the Western Free State began to think differently about soil rectifications as soon as our farmers began to plant soybeans. We're still starting out our soil sampling on a grid basis, know what's happening in your soil, lime your soil. And the different thing is we started seeing phosphorus and potassium rectifications as more important than we did previously because of the high uptake and the effective uptake of a soybean plant. This is just an illustration of what happens with 2 good maize yields and 1 very good soybean yield to your soil status. So before those 3 yields, the soil status of the potassium was 160 milligrams per kilograms. After those 3 yields, it's only 3 seasons and 1 season of soybeans, it dropped to 127 milligrams per kilogram. And the same effect with the phosphorus. So be aware, I don't think farmers can, especially in high yielding situations, can plant soybean crops without having soil samples or rectifying the soil. And yeah, this is just a practical illustration how we rectify the soil. You can see 2014 and 21 soil analysis, 2 soybean yields in between, around about 4 maize yields in between. And we can see the phosphorus status dropped in the high potential zone, and we rectified it accordingly on a precision basis. So the technology is here. Motivate your farmers to use those technologies to keep the soil status on par for the utilizations of all your crops in high yielding situations. Same with potassium. Just watch out. Potassium soil can only hold so much potassium, so don't over apply potassium. Maybe you'll get leaching because of the cation exchange capacity can't hold all that potassium. So in this situation, I rectify this soil to 100 parts per million because I know this soil can hold 100 parts per million of potassium. And it's on par for my maize fertilization program and so forth. And another option, if your soil, the farmer is happy with his soil, it's on par and everything, you can always rectify the soil if you have an above average soybean yield on your yield data. So you just, for practical example, you added 20 kilograms of phosphorus and 20 kilograms of potassium for a 2-ton soybean yield, but a farmer actually took off 4 tons per hectare. You can just subtract that from the uptake that the soybean had, and you can rectify everything that's taken out. And you can even incorporate your soil analysis. If you have a part in the field that had a high potassium status, you want to utilize that a little bit without cost, you can subtract that what you should have added or would have added from that. All right, then a summary. 10 to 20 kilograms of nitrogen for a starter is a good idea just to get that seedling strong, especially in the western part of the country where we have low organic matter, low residual nitrogen, and soybean can utilize build-up soils very well. So keep the soils in a good state that the farmer can have profitability from the soybean to the maximum. Balanced fertilizer mix makes sense. So if we refer to the 2 trials that we had mixes in, the soybean really reacted good on that. The guidelines is still applicable, and watch out for high yielding soybeans in sandy soil. It depletes it quickly. Obviously monitor your soils and rectify accordingly. Thank you.
Okay, any questions for Kobus? I have 10. I really do have 10. Okay, firstly, Kobus, farmers for a long time have been using ammonium sulfate at plant, yet I saw in the trial that you had, treatment number 8, I think was ammonium sulfate, and the effect in terms of SAT analysis was AB. So everybody's asking these questions. Ammonium sulfate at plant then seems to me to be a waste of time, or there's no ROI. Or did I misunderstand that completely?
No, I think I agree. Too much nitrogen with plant, 40 kilograms without phosphorus and potassium with it. I agree. It's not economically viable. Okay. But I remember it was 44 kilograms, so it was quite a lot.
Okay, so Corner again, remember in terms of claims and agronomy, one of the questions is what was the end status at the time in terms of the nodulation didn't happen? There seems to be a pattern here that we need to get hold of. I don't know if you want to answer that at this time, but there's a whole picture that comes out, but I'll leave it at that. The next question is carryover in terms of nitrogen from previous crops. Now, if you're going to be putting in soyas and the end status is quite high, if you looked at that one slide that Karin had, it was very clear that nitrogen and or molybdenum and or heavy metals, if I had it correct, is actually a no-no. Yet part of your recommendation is to go in with a quantity of nitrogen in that plant. This seems very contradictory. I don't know how you guys feel. I mean, we come to grips with this absolutely every day. Farmers are asking me what to do, what should they plant, is fertilizer plant available, what does it do? I don't know. Jonathan, perhaps you can also have an input here.
As I said, it's dependent on the area and the carryover from the previous crop. We, in the trial specifically, and in the western part, we a little bit of nitrogen that we see is positive, just to get a seedling strong, but too much obviously will give you a negative effect, have a negative effect on nodulation. But we balanced mix, a plant mix as such, usually gives us a good yield response and a good seedling. Because the thing is, if you have windy situations or so in the Western Free State, and that wind is blowing and you have sand damage on that young soybean, it's going to have a very negative effect on your long-term yield. So if you can get it a little bit stronger with a little bit of nitrogen and phosphorus and potassium, you usually get it faster out of the wind or faster out of the stress situation. And then we didn't see a negative effect on nodulation at all. As the first slide you've seen, actually the mix had a positive effect on nodulation because I had a stronger seedling.
Yeah, Jonathan, would you like to comment?
Yeah, I'd like to, please. Yeah, Kobus, thank you. That was really good presentation. Yeah, I think on the phosphorus and the potassium, I think when we talk about nitrogen, the nitrogen residue or carryover from wheat, for the sake, like in the follow-up schema, especially under irrigation, is quite a big issue. But there is a lot of stubble, unless they have the farmer rotation where they're actually burning, or they're not. If they're not doing that, it is a requirement. But there's a lot of carryover, especially in that high yielding situation where you're seeing the nodules formed far later than anything else. And also maybe a higher dosage of application as well. But I do agree with the 10 kilograms or 20 kilograms of nitrogen, in especially the sandier soils. I think it's really good. But the biggest one up, my question is really the potassium, because that's your key driver on soybeans, or phosphorus and potassium and sulfur. So how you be able to sort of prevent, because we're seeing in dry years where we put in KCl in the band, it is drying out that rhizosphere quite significantly because of the high potassium requirements. How would you go about that? Would you rather go to the broadcasting way, pre-plant with the broadcast KCl, because that would save you a lot of dehydration in that rhizosphere at planting?
Yeah, me and some of the colleagues, we talked about it. And the Eastern Highveld, for instance, is more of spreading the potassium out, because their soil and their plant row widths and so forth is applicable for spreading it out. In a Western Free State, we'd rather go into the band and put it away from the rhizosphere, the immediate rhizosphere, because we've seen the graph, it needs its potassium a little bit later. So it's fine to just not put it too close to the seed, but place it a little bit away from that. Like the 234 trial, for instance, it's 20 centimeters below where the seed was planted, so it's not in the immediate rhizosphere. Yeah.
So yeah, that's a great recommendation. Yeah.
Yes, Bridgitta?
Tom, thank you. Maybe if I can just ask Kobus, can you just go back to those results, the slides on those trials? I just want to point out something. We are all, sorry, I don't know why I've got reverb.
which one are you looking for, Bridgitta? The one with the 8 treatments? Ah, okay.
Oh, sorry.
That one's the 8 treatments. Okay, the 8 treatments, the graphs. Sorry, I'm not going to talk into this thing. Just look at treatment 2, 0N, 20, and 14. Look at treatment, what was 1, 2, 3, 4. Look at treatment 6. 21P, 7N, what's it, 0K. Look at the yield. I know we're all excited about the N's that we put in and the higher yield that we get, but just when you look at those two, what's it, 5 kgs extra. 3513 versus 3518 with 0N and 7N. Is that 7N really worthwhile if you compare those two? Yes, I agree. Adding N will increase your yield, but is that a responsible way of farming for the future when we know that N is a problem when it comes to environmental issues? That is what I'm trying to show, and that is where N will definitely have a detrimental effect on Rhizobia inoculation or nodulation, and I'll show that to you in my talk. But just look at those two. Is 7N really worth it? I think the problem here was P, and that was addressed with the 20 and the 21. Or am I looking at something different, Kobus?
No, I don't think you've missed it, Bridgitta. Again, please, just one trial doesn't make a summer, if you know what I mean. But certainly the indications are in terms of the stat analysis and the treatments. That seems to be the indication. This is why I wanted to come back to the ubiquitous question of in a plant, yes or no? If so, how much? And if so, in what form? Because again, we can go to the question of using KCl versus potassium nitrate or something else. And again, which form is going to be the best for the farmer? Karin, going back to your slide, have you got any information on that one?
Let me try this. I think what is really important is to understand that this is all very context dependent, and it depends on the environment where you plant, the current status of the soil, and I agree with you. We always forget that when you farm, you're removing a lot of nutrients from the area, which under native or natural conditions will just be worked back into your soil. So that needs to be replaced. And if you have a stronger plant that's less stressed because you get an initial little boost of nitrogen, then I think that would be better at the end of the day. But if you maintain high nitrogen levels, I don't think you're gonna have struggles with nodulations, and then you'll have to add all the nitrogen that's required for a good yield, which I think is going to be quite costly.
Yeah, that definitely seems to be my information as well. So I see you nodding big time, Jonathan. I know you've bumped your head against this a few times, but so have I and a few other people. I think we have to be very careful when we start recommending nitrogen at plants with soya. Again, with proper trial work, with proper information in a proper area. I mean, every farm is so different to another farm, even the one next door, even differences within that. But to ubiquitously go in and say that you're going to apply an amount of nitrogen, and with the negative effect that we know that the carryover in terms of rhizobia and or nodulation, that trade-off is exceptionally dangerous. I don't know how you guys feel, but I'm sure, you know, the 40, 50 years I've been around, that's been an issue, and it's exceptionally contentious. Jonathan?
So, Tom, I agree with you. I think the benefit of the nitrogen is in some processes, I've seen some really in the Western Free State, Northwest, I've seen some significant results with the nitrogen, especially at the planting, but that 10 to 20 kilogram. But, you know, moving over that, it really reduces the yield. But I just think the salt contents in the band is the biggest issue that we are facing with nitrogen and potassium, because these are the two elements. And if we're not putting them in the band, especially in a dry year, we had quite a bit of guys use MAP or
certain amounts of fertilizer inside in a pop-up mixture in a liquid band application, and that really negatively influences the rhizobia because of the salt concentration in that concentrated form. But I mean, if you are looking at, we do have low organic matter, and sulfur and nitrogen are really important in those Western Free State areas. So a little bit of nitrogen does definitely, in those areas, in those situations, it is farmer by farmer, but or block by block, but I've seen some great results. Yeah, so I would rather be more worried about this total salt contents and how do we mitigate that? And I think Kobus, with that deeper application of the potassium, it was significant. And if we could do that, in the let's say Mpumalanga, the Eastern Highveld, where we're broadcasting the potassium chloride, it's really helping us a lot in the dry conditions. If we have a wet condition, I don't see the salt being that big an issue, but in dry conditions is the major, yeah.
Yeah, that seems to be the answer, Jonathan. Thank you. Oh, lots of questions out there now. Okay, yes, sir.
Kobus, ek het meer 'n praktiese vraag vir jou. In terme van die toediening van kunsmis. Daar's 'n paar boere wat rolstaalde gebruik om dan hulle MAP's helemaal te bandblaas. En ek het gevind dat van die rolstaalde lig daai stroom kunsmis lig op en is dit dan binne in die plantsone. En my vraag is, ek het nou gekyk na die ene, as dit nou maar 50 kilogram MAP is dit baie min. My vraag is, die soutte in so 'n band, sal dit 'n effek hê op die saailinvestiging?
Ek dink definitief, maar byvoorbeeld MAP het 'n baie lae soutindeks en dit hang af hoe die rolstaaf hom natuurlik versprei, nê. So as die rolstaaf daai band direk opbring en jy gaan in daai band plant, dan is dit 'n moeilikheid tot met 50 kg MAP, maar gewoonlik kry 'n ou laat hy hom tog 'n bietjie versprei en ons het nie regtig daai moeilikheid nie. So maar as hulle begin kalium bysit en so voorts, dan weet KCl wat hoë soutindeks het, is dit definitief 'n risiko. Ons het al gesien die soybean saailing is baie sensitief, of die saad is baie sensitief vir 'n klein bietjie sout.
Ja, Kobus, ek wil ook net vra net, so daar by die derde een, daai 13-17 en die ander. Jy het nou gesê julle het jy het eintlik gepreplant. Dit was nie geband of was dit gebandpas of was dit gepreplant? Jy het gesê die 234 was gepreplant.
Ja, die 234, die 234 was 'n preplant mengsel. Hierdie een spesifiek was plantermengsels geweest.
Want ek dink dis waar dit inkom en dis wat almal nou oor praat, nê, is daai soutindeks, nê. Ek dink daai kaliums raak net te hoog.
Ja.
So jy moet pas op. En ek verstaan hoekom julle dit in die Wes-Vrystaat doen, want dit help nie jy strooi dit uit en dit bly in die oppervlak en is droog later in die seisoen nie, want jy wil dit in die profiel dieper hê. So ek dink 'n pre-plant aksie kan dalk daai oorbrug.
Nee, definitief. Jy kan basies hierdie as 'n risiko vat met hierdie hoeveelheid vir 'n plantermengsel. Dis 'n proef en alles. Maar as jy hierdie hoeveelhede wil insit, moet jy definitief jou kalium wegvat daarso. Dit is so, die soutindekse is 'n bietjie hoog en die proef was al herhaal in droër jare en dit het die stand benadeel. Ja.
Okay, any more questions? I've got one or two more. The comment was made in terms of doing liming. Now, for the ag chem guys amongst you, you all know what happens when you start doing liming opposite certain ag chemicals, such as atrazine, the triazines. What happens? Can somebody tell me? Come on, you guys are clever, I know many of you. Okay, let me refresh your memories.
Atrazine becomes more available.
100%.
Can you lock in and if you apply light, it becomes available if it's still a crop.
100%. So that's the bottom line. When you start doing liming, be careful when you're going after maize or something like that. And then you start saying, okay, we have to do a lot of liming because you want to correct the pH going into soya. When the triazines start becoming available, they're going to kill those soyas. And a lot of guys think that's a lot of nonsense, but it really is like that. So be careful when you're going into a rotational system in terms of the previous year, in terms of the triazines that you're going to put down, because they're going to be released. And a triazine, when it comes to soya, is really not a good thing. I'm sure you guys know about this. So I just wanted to point that out in terms of from a cropping and an agronomy point of view, Kobus, that comes off the issue is.
Yes, we've seen it especially on nodal situations.
Yeah.
Your atrazines will stick on that compaction layer, and obviously your root will grow there as well, and then usually you get a negative impact there.
One last question from my side. In terms of molybdenum, there's a lot of controversy around molybdenum and applying to the seed at plant versus applying sodium molybdate and or another molybdenum source as a foliar applied product. Now, the clever guys here will tell you that when it comes to rhizobia as an organism and contact with a heavy metal, that generally is a no-no. Yet there is still a persistence when it comes to some people. They like to apply some form of sodium molybdate and or whatever it is, a molybdenum-containing product on the seed at the same time as the rhizobium. Maybe you can talk to that. I'm not sure if you're going to cover that with your talk, Bridgitta. Not sure. I'm sure you can. Maybe you'd like to comment on that, Kobus?
Yes, obviously the guidelines out there says sodium molybdenum is toxic to the rhizobium. Practically, my farmers don't add sodium molybdenum to the seed as such. We'd rather put it all in furrow, but also very, very low quantities, and we'd rather go for a foliar very early in the life cycle of a soybean. But I know there is guys that have success with adding it and planting it out immediately at a very low rate, lower than that is subscribed. Like I think it's 30 milligrams per 100 kilograms of seed. Yeah, but the guys go to 30. So yeah.
I've got a question. Jonathan, you talked about cobalt, which is very important in the bunning process. How does the cobalt get applied? Does it go with the rhizobiums or the inoculants? How do you do that thing? Very important also because cobalt is one of the minerals, products, substance of concern at the GFS. So that will have to be cleared by Act 36 very soon if you want to carry on with that. So perhaps anything about that?
Yeah, definitely. Cobalt's also just as important, or maybe not as important as molybdenum, but in smaller quantities than molybdenum. But it is also just as important for reduction of ethylene within the plant, but also increasing nitrogen fixation and the nitrogenase activity of the plant. So generally we would apply the cobalt as with the molybdenum as a foliar application. As soon as possible after planting or after emergence from around about V2 to V4 stage would be your optimal. You can apply it in the soil, but your pH plays a huge role in the binding of these metals. So for example, the phosphate, if you've got a nice high phosphate, your molybdenum or your molybdenum will be bound by that phosphate, and that's also a big issue why the infra. Sometimes, if you have a good pH, it's good. But if your pH is low, significantly that you wouldn't be able to apply molybdenum with success because of the binding it to the phosphate. So we generally on the cobalt side we would apply that with the molybdenum at V4 and maybe up to R1 stage.
Thank you very much, ladies and gentlemen. Okay, we'll call it quits there. Kobus, thank you very much. Well done. Okay, can I have you all back here at 11:45, please? 11:45. Thank you very much.